What if I told you that the biggest challenge in biology today isn't happening in a wet lab, but inside a computer? Today, we're diving into bioinformatics. It's the science that was pretty much born out of necessity when biology suddenly had way more data than it knew what to do with. We had to learn how to read the instruction manual for life itself.
So picture this, it's 2003, and scientists announce one of the biggest achievements in human history. They've sequenced the entire human genome. Absolutely incredible, right? But this huge success created a brand new, kind of unexpected problem. We had finally been handed the complete book of life, but we had no idea how to read the language it was written in.
And this wasn't just about us, about humans. All over the world, sequencing technology was just exploding, generating this absolute tsunami of genetic information from thousands and thousands of other organisms. All of a sudden, biology was literally drowning in data. And the old way of doing things, a single scientist at a microscope, it just couldn't keep up, not even close.
And that really brings us to the heart of the matter, biology's big data problem. The challenge wasn't about getting the information anymore. It was about figuring out how to turn all that raw data into actual, useful insight.
To give you a sense of the scale here, just think about the common house mouse. Its genome is about 2.5 gigabases long. What does that mean? Well, if you were to print out its genetic code in a standard font, the book would be taller than a 30-story building. Now just try to imagine doing that for millions of species. It wasn't just a data problem, it was a full-blown data crisis.
So, faced with this absolute deluge of data, biology had to adapt. It needed help from a field that really knows how to handle massive amounts of information. So, out of sheer necessity, biology and computer science teamed up, and a brand new science was born.
And that field is bioinformatics. The best way to think about it is as a bridge that connects biology with computer science, with math, with statistics. It's the discipline that uses pure computational power to pull meaning out of the molecular data that defines life itself.
Now, this story isn't just about machines, it's about pioneering people. And you have to meet Margaret Oakley Dayhoff. She's often called the mother and father of bioinformatics. See, way before the big genome projects, back in the 1960s, she had the incredible foresight to start collecting all the known protein sequences into the first ever bioinformatic database, her Atlas of Protein Sequence and Structure. I mean, she was building the library before most people even knew there were books to put in it.
This timeline really shows you how it all happened in parallel. You've got 1953, we discover the structure of DNA. Just a little over a decade later, Dayhoff starts cataloging what it makes. Then by 82, public databases like GeneBank pop up to handle the data flow. And then bam, 2003, the Human Genome Project just opens the floodgates. Every major biological leap forward basically demanded a new computational leap just to keep up.
So what do bioinformaticians actually do? Well, this next section is all about their toolkit, the core concepts, and the software they use to, read that instruction manual for life. Okay, first things first, we have to understand how information flows in a cell. It's called the central dogma of molecular biology. Your DNA, that's the master blueprint. When a specific job needs doing, a temporary copy of that one instruction, called RNA, is made. And then, that RNA is read by the cell's machinery to build a protein, which is the actual little machine that goes and does the work. Bioinformatics tools are designed to analyze the sequences at every single step of this process.
So let's say a scientist discovers a brand new gene sequence. How in the world do they figure out what it does? The absolute easiest way to think about it is like this. It's a Google for genes. You take your unknown sequence, that's your search query, and you use these super powerful tools to scan a global library of all known biological information, just looking for clues.
And here's what that Google for genes actually looks like. First, you have the databases like GenBank. These are the giant digital libraries. Then you've got the search algorithms, like BLAST. These are the powerful engines that actually comb through all those libraries. The result might even be a 3D structure from a place like the PDB, which lets us literally see the tiny molecular machines we're studying.
So what are these search engines looking for exactly? The answer is a really important concept called homology. It's this core idea that if two gene sequences are really similar, it's almost never a coincidence. It means they probably share a common evolutionary ancestor. Finding a homologous is kind of like finding a long-lost cousin in the genetic code.
And homology comes in two main flavors. First up, you have orthologs.
These are basically the same gene, but in different species. A great example is the gene for alpha hemoglobin in a human and in a mouse. They both came from a single ancestral gene, and they still do the exact same job. Then you've got paralogs. These are related genes, but they're within the same organism. This happens because ages ago, a gene was accidentally duplicated. So now a human has genes for both alpha hemoglobin and beta hemoglobin. They're related, but they've evolved to do slightly different, specialized jobs.
Now, understanding these relationships isn't just some academic exercise. It has huge real-world payoffs, from figuring out diseases to literally mapping the entire history of life on Earth.
You know, for decades, finding new drugs was basically a game of trial and error. Scientists would have to test thousands and thousands of compounds, just hoping one of them would work. It was an incredibly slow, expensive, and honestly, pretty inefficient process. Bioinformatics completely flips that script with something called computer-aided drug design, or CAD. Instead of guesswork, it's a smart, rational process. First, scientists identify a single protein that's critical to a disease. Then, they use supercomputers to digitally screen millions of potential drug molecules, looking for one with the perfect shape to fit in and block that protein. They can do years of lab work in a fraction of the time, all before anyone even picks up a test tube. And beyond medicine, bioinformatics is our time machine. By comparing all those homologous genes, those orthologs we talked about, across hundreds of different species, we can actually measure how similar their genetic code is. And this lets us map out the tree of life and reconstruct the evolutionary paths that connect every single living thing on this planet.
This chart gives you a pretty clear idea of how it works. So remember orthologs, the same gene, different species? As you'd expect, the alpha hemoglobin in humans and mice show very high similarity because they do the same critical job. But the paralogs, those duplicated genes inside a human, have had more evolutionary time to drift apart. So the alpha and beta hemoglobin are less similar. And that difference in similarity is exactly what scientists use to tell the evolutionary story.
Okay, so finally, let's look ahead. Bioinformatics gave us the amazing ability to read the code of life, but now it's laying the foundation for learning how to write it.
And the future here is just incredibly exciting. We're talking about things like pharmacogenomics, where drugs are literally customized for your personal DNA. Systems biology, where we can build digital twins of our own cells to predict how they'll react to things. We can start pinpointing the exact genetic roots of diseases, and even design crops that are more nutritious and can survive droughts.
What's become so clear is that bioinformatics has totally transformed biology. I mean, it may have started out as just a tool to solve a data problem, but it has evolved into the fundamental language we now use to ask and answer the deepest questions we have about life itself.
Having the power to read and understand life's source code is, well, it's incredible. And it leaves us with the ultimate question. As we get more and more fluent in this biological language, the real challenge isn't just technical anymore, it's creative. What new stories will we choose to write in the book of life?